Okay, everybody, this is Dr. Ed Park from Recharge Biomedical, and today we're going to be discussing aging. Um, Above me, we see what looks like a leaf. It's actually all the roads that lead to Rome or away from Rome. And the reason I bring this up is because um, that aphorism in that kind of contains the problem. Basically, we're all getting old. We're all getting sick. Eventually, we think we're going to die. And when you look at the subject of aging, everyone has their own pet theory. There's one way to get to Rome, but in the truth of the matter, there's many ways to degrade an organism, its function, and there's many different theories that compete to the detriment of wisdom. So a lot of things can be true simultaneously. They can't. You don't have to think of it in one axis. Initially, about 13 years ago, when I started taking the telomerase activator, I got excited about the telomere theory, which I think is one main artery into Rome. And I'd like to encourage you, if you're interested, to study the diagram there in the middle. That octagon shows that short telomeres have been correlated with disease in many different, perhaps almost all different diseases. So this is uh, a common denominator, and it could be driving a lot of the aging we see. First book I wrote, self-published, was called Telomere Time Bombs, and I talked a little bit about my theories there. And then the most recent book in 2018 was The Telomere Miracle, which is more of a self-help book about the science of increasing telomerase activity. And as you might predict, things like better sleep, exercise, better eating, better thoughts even help to slow aging, which makes sense. So today we're going to talk about what causes aging, in my opinion. If you want to go deeper into a dive, I did make an exosome video, and you can find that at tinyurl.com/exosomes2. And then finally, we'll speculate as to how exosomes could mitigate aging, if at all. So the analogy I've come across, I think works best, is the Hindu Trimurti, and of course, most of the languages we speak are Indo-European. So Tri is three, and Murti is form or morphology. So in their pantheon, they have Brahma on the left, who is the creator, Vishnu, who is the preserver, and Shiva, who is the destroyer. And these are not given the sort of negative or positive valences that, let's say, the Abrahamic religions say, well, Satan bad or Yahweh good. It's not like that. But we do look at this next aphorism, which is if all you have is a hammer, everything looks like a nail. And of course, to the detriment of our greater understanding and survival, a lot of scientists say, well, this is my pet project. Now it's going to be resveratrol. Next week it's going to be a sirtuin inhibitor called NAD. People get on one trick and they pay their mortgages and their boats and their colleges. But the point is, this is very, very complicated. There are certain things that are true-ish, but the integration, the whole system is very complex. So again, if we use the analogy, Brahma will be like the stem cells. And it's a truism that we are made of stem cells. We started as one stem cell, the fertilized egg. We made 7 trillion, and we whittle away and we survive and thrive for 80 something years usually on that one cell. So we'll have to talk a lot about stem cell ecology. Vishnu is the preserver, and of course, the sine qua non of being a stem cell is being immortal. And so being able to preserve is something that I've been partaking in for 13 years. That is the telomerase activator. If you'd seen me 13 years ago, it had gray hair and reading glasses, and I do believe it's kind of flattened that curve. Are there some downsides to that? Perhaps. But again, it's not good to think of everything as a hammer and a nail. Finally, something that's emerged in the last few years is called Shiva or senolytics or destruction. So what we talk about, there was just a trial about 4 months ago that failed through Unity Biology or Biotech, this famous guy, Ned. Anyway, the point is they're throwing money at something. I don't think it's as good as it could be, but the principle is sound. In other words, if you have old cells and you kill them, maybe young cells will replace, but we'll get a little more into that later. About 4 years ago, I was pleased to be invited to UCLA to listen to some lectures, a few hours of lectures by the original scientists. Of course, my friend was the man who invented the sort of CPG replacement technique for epigenetic measuring. And so he is collaborating with Dr. Steve Horvath, who is a scientist leading the epigenetic clock. And I think it's worth it to talk about what the epigenetic clock is and isn't. On the one hand, it could be data mining. In other words, what they do basically is they look at these sites and they categorize them, and then they reverse engineer a model to say this is highly correlated with aging. And the results are quite impressive. I sent my blood from the same draw as two different people, and they nailed it. Yeah, unlike some of the other tests with telomeres, sometimes they use age as a proxy, which is cheating. But I said I was a 27-year-old and they nailed me as a 41-year-old. And I said I was a 51-year-old and they nailed me as a 41-year-old or whatever. So there is a high correlation, but is it just data mining or is it causation? A lot of people jumped on the bandwagon and again, they got their hammers and they looked for nails and they said, oh my God, causation, of course. Well, we don't really know because it's very important um, to distinguish the two. It could be both in some instances, but if we look at this published paper from above, this is just this year, they looked at a bunch of mammalian species and they found that there were 13 common sites of methylation. And methylation is when you add a methyl group to the DNA so that the gene is less expressed or shut down. And it suggests that there may be 13 common sites across all mammalian animals that age. And so there may be some causation, but in the main, I think this middle point is very interesting. And it goes back to the first point. During the lectures, they looked at progeria. Steve Horvath, good scientist, he looked at kids with premature aging, and they didn't have advanced epigenetic clocks. So that really throws a monkey wrench in the theory that it's causal, doesn't it? But the epigenetic clock, I think, when I sat in the boardroom with the people who invented the test, that I asked them and they kind of sheepishly looked aside and they said, well, it's not like DNA. DNA copying is so good, you make an error on the order of 1 every 4 times you try. I mean, that's like, I don't know, 8 billion letters being copied perfectly, whereas epigenetic software copying is not. It could be lossy, it could be gainy. Of course, if it was slightly gainy, it could be like the accumulation of errors. And so that might be something so trivial as just the epigenetic copying is not that good, so that's why we get old. This central cartoon is very important to understand, and what it basically explains is stemness. So, as I said, we started as one fertilized egg. We ended up in 7 trillion cells that are surviving 80 something years. And the way we do that is by establishing stem cell niches. So, in your retina, in your pancreas, everywhere in your skin, your hair follicles, you have cells that are stem like. And stemness is comprised of two general things. One is immortality, which is conferred by telomerase activity, and the second is asymmetric division. Very important. It sounds like a lot to think about, but it's very intuitive if you think about it. A mother has to divide into another mother and a daughter. That's the kernel, that's the crux of what stemness is. Uh, a mom makes herself immortal. She copies asymmetrically. She makes a more differentiated daughter and she continues on forever. And that's how we can be so relatively immortal. Now, what if we had different set points? What if our clocks were set differently? Of course, we understand that the human life cycle is basically in utero. You start from that one little cell, we divide rapidly. There's a lot of potency. In other words, these cells can become many different things. And as in life, as we go along, there's fewer possibilities, perhaps. So the cells that we use are adult, even though they come from a newborn baby. And that's because the adult stem cell doesn't have this ability to become a germ cell. In other words, it's not omnipotent, pluripotent. It has some potency, but they're adults. So when we talk about germ cells, these are sex producing cells or baby producing cells, eggs, oogonia, and sperm, spermatogonia. And these are kind of immortal. As we go through life, there are major events like getting out of the uterus and then getting out of the home with our puberty. And so, of course, the end of puberty marks the end of growth. As we get older, arguably men might have a diminution gradually of their hormones, but women have that drastic shutdown, and that can accelerate aging as well. And then finally, I was talking to the guy who proposed the idea of a telomerase, and he really believes that there's some kind of gravitometer in the inside of the brain, which measures how many moons you've gone through, and it keeps a kind of like a prisoner, keeping it etching on a wall, how long you've been alive. These are all great theories, but the fact of the matter is, if we look at this chart of giant humans, you know, allegedly there were evidence of giant humans. Whether you believe that or not, I don't know. Um, Many of the folklores talk about days of the giants, but of course, if you tweak the parameters of when puberty happens, or you know, how that program is initiated, we can get relatively larger people. Achondroplastic dwarfism is that's shutting down too soon, and above me we see where the growth plate happens. So the longer you grow, the more efficiently you grow, the taller you get. So it's not absurd that we could somehow figure out how to reset that and make people taller or smaller. So that's an interesting point. Let's get a little bit far afield here with immortality. So we see Jules Verne, arguably the father of speculative sci-fi, and he's looking at a giant squid and the Nautilus. Okay, so we know there's giant invertebrates. We know there's giant jellyfish. There's giant squid. Squid, allegedly, a lot of invertebrates have a high percentage of stem cells. They keep growing. Unlike ourselves, we have to fit into cars and houses. We don't really grow, even though that's teleological thinking, after a certain point. But we know the squid is effectively immortal. It has so much stemness throughout its body, and it doesn't have the limitations of relative disadvantage as we would if we become 10 feet tall. So they grow in the ocean, they grow big, then they're kind of immortal. To the left, we see the bristlecone pine. As every time I drive to Mammoth, there's the bristlecone pine forest. I've never visited, but these trees routinely live 3 to 6,000 years. And according to this study, they have a long telomere length and high telomerase activity. So that's part of their secret. To my left is Irv Gordon and his Volvo, which he drove for millions of miles. And, there's no secret there. He just replaced the parts. And so they say that our bodies are constantly replacing their parts. Luckily, we are self healing and we have wonderful mechanisms. But so we carry our replacement parts around with us. So, you know, I'd much rather be a human than a Volvo because we have so much more latent potential to create new parts. All right, let's go into the real sort of crux. This is probably the most important slide if you want to know what causes aging. So if you want to know what causes aging, go back to Mother Nature's model. Look above me on this adapted chart that I adapted. In the bottom half, the telomeres are not shortened. These are things like Ehlers-Danlos syndrome, which is a collagen vascular disease, a Bloom syndrome. The three in the middle are problems with DNA transcription, we now know. So you can kind of get older or look bad if you have these, but I wouldn't consider these classic progeric diseases, meaning accelerated or towards aging. Above the line, I would consider progeric. And again, we have this theme of DNA repair is messed up with ataxia and Nijmegen breakage syndrome. The helicase, which is the winder and unwinder of the DNA that's required to copy, that's messed up in the blue, Rothman, Thompson, and Werner. Hutchinson-Gilford is a classic one that affects the scaffolding that you need to set up the replication of DNA. But the most classic premature aging syndrome is dyskeratosis congenita. We touched a little on this in the alopecia or the hair loss webinar. If you look to my left, these kids are bald, gray and bald. So this is one of the hallmarks of aging, of course, we know. And it's highly related to telomerase activity, as we'll see shortly. Again, the best theory, I mean, everyone has their pet theories that they write books and give lectures, but the best theory is the most obvious one. It's immediately accessible to all of us, which is that we require stem cells. And this Dutch lady to my left came out, I think there was a study 1 or 2 years ago. They looked at her blood producing cells and they really only came in two varieties. Now, you talk about varieties of blood producing cells, there should be many, many because of the rearrangement. But the clonality showed that she really only had two versions of stem cells, whereas we might have thousands or millions. So, like a cell phone battery that is charged, she was discharged at 115. She only had two clonal variants. So, throughout our lives, both in the local niches, hair follicles, either retina, pancreas, but in the bone marrow, which is critical for that immune system, we get a depletion. And why do we get a depletion? Why aren't they immortal? Because again, with that cartoon that we see in the middle, mama makes mistakes. Mama can only be as good as her last mama. So if there are DNA errors, if there are epigenetic shutdowns, that's bequeathed to the next mama. So gradually, she becomes a less perfect version. She becomes a further boondock road out from Rome. We all become chimeras. The entropy is introduced both into our genetic code and into our software epigenetic code. So we become older because the Brahma is gone. The pure stem cells are deranged. They're depleted and they undergo differentiation. And what that means is depletion is because of differentiation. When a mama fails to do that little trick, which we don't really understand how she makes a perfect mama and a daughter, instead she goes to a less perfect mama, which is shown in that intermediate pluripotent cell at the bottom of the branch, or she could go into two daughters and just lose her stemness, lose her telomerase, or lose her asymmetric division. So the nitty gritty of what asymmetric division is and how it's tweaked is really not known yet. Hopefully it will be someday. And we see why this Dutch 115-year-old lady got old. You see all these wonderful things: platelets, red blood cells, the immune system, the granulocytes that we talked about last week in the autoimmunity lecture. They're all depleted. So you have no defense against your own cancer and autoimmunity and infection. It's not uncommon that people like Jack LaLanne with healthy bodies, they just get overwhelmed by a cold. All right, so getting old is a depletion via, most likely, inadvertent differentiation of our stem cells. So how would we mitigate this? Well, when I was giving a stem cell keynote lecture in Korea a few years ago, I was touring one of the state-of-the-art clinics, and for rich 20-something-year-olds, they're banking their blood, meaning actually it's bone marrow biopsy. You can just bank it. Now, it's arguable whether these cells survive after cryopreservation, but you can get a record of your most perfect 21-year-old self and store it. And so why would you do that? Well, we see this graph of what the global stem cell banking market is. Um, we it grew from 2018 from 6 billion, and they project it doubling roughly by 2025. So the idea is a good one. You take your healthy version of yourself before you become degenerate and differentiated and gene silenced, and then someday in the future you can thaw some of them, and you can then replenish, recharge, and that makes sense. Of course, I think this is one of, I'm not privy to the exact intellectual property, but our exosomes again seem to come from one placenta, and this is kind of the technique I believe they use. They thaw out the master mother batch every few months and get a fresh batch, but it's still day zero, and so you could do this for yourself and replenish. You could clonally expand millions of stem cells and just keep on going that way. Um, Another great breakthrough in science was a few years ago. They had this Yamagata or I always forget, but Yamanaka factors induced pluripotency, and he found that with the introduction of just four proteins, or four messenger RNA, if you will, you could cause any cell to de-differentiate back to a pluripotent cell. Uh, what's shown in the cartoon to my left is that when you do this, you have the OCT4, SOX2, KLF4, and arguably the fourth one is c-MYC, which we talked about in the hair loss, Yamanaka factors. When you do make a zombie cell or make a differentiated cell back into a mama stem cell, a lot of these just die off because there's something wrong with their DNA. The P53, which is the watchman, the policeman, will cause that induced stem cell to die off. So if there is no P53 activation, then you do get this holy grail, if you will, of induced pluripotency. Why is induced pluripotency good? Again, I don't think it's good. I don't think it's something that we will use, but it does prove a very important principle, which is that cells not only lead away from Rome, they lead back to Rome. So you can get de-differentiation, you can get younger behavior, and I believe that's kind of where we're heading with these exosomes. In addition, there could be matrix host factors, although I don't really know. There could be something about the way that the bone marrow is and the atmosphere or the milieu that is not so good for growing healthy or maintaining healthy stem cells. So as I said, most of my experience on YouTube and writing has been with this telomerase activator, which is kind of a misnomer. There was one great study by Mulgara which looked at the mechanism, and was it a direct telomerase activator in terms of gene expression? No. So it could be a downstream effect of what's called the MAPK pathway. Um, so what we're basically tweaking with is all these protein signaling pathways, cell differentiation and signaling pathways. But the end result could be stabilization, relative lengthening to a point of your telomeres, not unopposed. If you want to learn more, again, pick up my book, Telomere Time Bombs, and then if you want to know how to tweak it, The Telomere Miracle. And what's shown there is the analogy I came up with two trapeze artists. So basically, when the telomeres shorten, the ends become sticky. The machinery doesn't know that they're not just cut chromosomes, so it sews them together. The next time the cell divides, the trapeze artist will be ripped apart at different areas, and that's the engine of what drives mutation and probably depletion and even things we don't like cancer. But luckily, P53, as I explain in the books, is around to kill off any cell that misbehaves. They suicide. Why do I say that dyskeratosis is the ultimate aging model? Because what we see here is that in the cartoon to the left, there's three major players here. There's the TERT, which is the RNA transcriptase, or reverse transcriptase, I should say. The TERK, which is the sort of RNA component or the template for printing new um, telomeres in 6 base pairs. And then there's the dyskerin off to the right. It's kind of cropped, but it's called dyskeratosis congenita because we don't grow keratin, things like nails and hair, well. But this molecule was even named dyskerin, so it's very well described. And what we see is that you can mess up any of these three things: the dyskerin, which is like the keyhole, the TERK, which is the key, and TERT, which is the engine, and you get various forms. Anytime you mess up one copy of any of those three things, you get the same syndrome of premature aging. You can be autosomal dominant inheritance, autosomal recessive, or X-linked. So we know that this is a very robust model for premature aging. And finally, if we look at the Trimurti, there's the birth of the stem cells, the maintenance of the telomerase, and then the Shiva of the destruction. And again, we see this P53 again and again. P53, same diagram to my far left. When you induce pluripotency, if there's a problem with it's usually what's called uh, there's a point at which the chromosomes line up, and if the number of tethers is wrong, it says, "Uh-oh, we have the wrong number of chromosomes. Somewhere we got messed up," and that signals P53 to induce suicide. It pokes holes in the mitochondria, and it literally melts from within with that battery acid. So again, the aphorism is be careful what you wish for. The presumption, which may or may not be true from these early animal studies, is if you have an efficient senolytic or something that kills old cells, you're going to get younger cells, which has been shown in some of these mouse models to get younger coats, younger behavior, more hormones, less insulin resistance. But again, be careful what you wish for. We see the leaky pail. If you make more holes in the pail, do you have new fresh water to add in? And that, again, is kind of what I forgot to mention with the earlier slide, the home birth. I was an OBGYN, and home births are not that hygienic in some ways because, of course, there's stuff that comes out as the baby's head is pushed through the pelvis. But of course, if it's done with circulating water or, arguably a goldfish net, you can make it work. But again, the whole thing about going from one cell to 7 trillion relatively dysfunctional cells or functional cells, depending on how you want to look at the pail, as mostly empty or mostly full, we have the challenge of if we kill off old cells, do we have young ones to replace? And that, in the case of the 150-year-old Dutch lady, was a problem. If you did senolysis on her, she'd be dead. If you did it on a 21-year-old, maybe you get a real spring cleaning, a real car detailing, you'd be great. So be careful what you wish for. Again, above me is one of the most promising candidates of senolysis, is FOXO4 DRI, which I have experimented a little on myself. What is FOXO4 DRI? Well, we look at P53, and the scientists have found out that there's something called FOXO4, which puts a brake on the P53. The P53 gets gummed up, and it can't exercise that death program that we see to the left, the Yamanaka factors. If you do induce stemness and all the superpowers, if P53 is activated, it'll die. But if it's gummed up by FOXO4, it won't die. So the ability for us to get old is a good thing. If your cells can't get old, then you can't get old. If you couldn't get old, then you would die young. So again, it's kind of a rub. But what they did was a very clever thing. They created FOXO4 in a DRI version. Amino acids naturally are levo-rotated, meaning they have a left chirality. If you remember organic chemistry, there's like mirroring and yada yada. But the point is, they used artificial amino acids. So the sequence was the same in the FOXO4, but the some of the amino acids were dextro or right rotated. So it fit the keyhole. It competitively bound to where the FOXO4 was going. And like the classic drug model, it caused a decreased activity of the native FOXO4. And that allowed cells to die. Cells died, and then the phenotype of these mice got younger. Yay, exciting. So we finish up with my mom, who's ironically, her name in Korean is Youngju, and she feels younger, and she's 83. Um, of course, FDA has not approved these to treat aging. Because I'm her son, she believes in me, and because she's seen results, she's a real believer that she will maintain health and well-being. She's received 13 treatments. She was my original sort of guinea pig. And over the course, she's gotten two, I think, nasal treatments. We've done her knees, which no longer hurt, her shoulder, which is almost 100% now. We're going back to do her hip. We've done her hands for arthritis, as we talked about last week in the rheumatoid. So, it's kind of like refurbishing her Volvo P1800. But I think the most interesting thing was the day after the first IV, she said, I feel younger. I look younger. And I drove up and I saw her, and indeed, the energy she was sending out was different, which I think her memory is better as well. We were driving 2 weeks ago, and over this phone came a customer service, and they gave us a 7-digit phone number, and she just boom, remembered it. She couldn't do that a year ago or 2 years ago. I can't even do it now, 10 digits. So her energy is better and her optimism is better now that she has less of a fear of getting old and decrepit and demented. She wants to go back to medical school. I have to remind her she doesn't have the requirements, but she wants to rent an RV and drive the lower 48. I mean, just that kind of excitement, I think it's innate in her personality, but she just feels more optimistic. Um, I think that's also related to the HRT she takes. She's a big believer, as some of the older endocrinologists were, at DHEA, which is a weak androgen or steroid precursor. She has been self-supplementing with estrogen and some low levels of testosterone as well. So she has a real strong desire and belief that she can live. And if we're being honest about what aging is, I know if I forget or stop taking my telomerase activator for a few days, I get grumpy, and every day feels like day X plus one. Whereas when I take a telomerase activator, it feels like Groundhog Day, same day, which is its own sort of curse in a way, but I don't feel like I'm getting that much older. So just to wrap up, for people who don't know, exosomes are the product of all cells. My son asked me last night, well, are exosomes good or bad? If you take exosomes from a person succumbing to breast cancer and give them to a healthy woman, uh, the healthy breast tissue will show transformation, or meaning there's bad stuff in those signals. Whereas if you take exosomes from someone who's overcoming liver cirrhosis, then they can help heal a person who's suffering from liver cirrhosis. So the messages can be good or bad. The messages are tiny. They're 1/300th, 400th the diameter of a regular cell, and they're released in these bundles or packages as shown above me. They're simple 100 nanometer spheres, just like the coronavirus, just like I don't know what the size of the corona vaccine is, same technology, a spherical lipid bilayer with a messenger RNA in it. But in this case, we have messenger RNA, we have proteins, some lipids, and a microRNA, which are probably the big hitters. MicroRNA block transcription of certain genes, and as we talked about in the nerve webinar, can cause dedifferentiation, which is sort of that magical thing that we're all looking for. They're not cells. They don't have the machinery to copy DNA. They don't have machinery to produce energy. They don't have any of that stuff. It's just a message. And they don't have self antigens on the surface, so they can't be rejected by your immune system, which is very, very important. So as we finish up, we can speculate how this would work. Of course, exosomes are known to be very anti-inflammatory, as we talked about last week. They make the macrophages behave less inflammatory and more into a regenerative mode. The T cells are from Th1 to Th2. There's increase in Tregs, T17. All this stuff is kind of above everyone's head, except for a few people who can understand it. They probably improve cell behavior and communication. They reduce autoimmunity. They can regenerate stem cells both in the local niches, like we talked about the patient with the hair growth, uh, also in the hematopoietic cells. So your immune system in the long run will get better. In the short run, if you got a corona vaccine, I was talking to the chief scientist and founder of Chimera, and he said, don't take it if you're getting vaccinated because you want that 14-day immune response. Um, we have possibility of dedifferentiation. Again, go back to the nerve regeneration webinar if you want to learn more about that. We can get apoptosis and replacement of certain cells. We can get a down regulation of the autonomic state, which again means that we're less tense and nervous, and we can get a reactivation of the endocrine axis. I've had several people tell me they felt more aggressive or their libido went significantly up. So there could be something going on there, both in the hypothalamus, pituitary, and organs. So if you have questions, you can email me at drpark@rechargebiomedical.com. There's a plethora of information at my website. I'd like to close with this wonderful movie called The Age of Adeline, where because of some freak accident, she had a telomerase mutation that she couldn't get old, and it portrays her living a life of, a benign vampire. She lives way, way beyond 115 years or something, and she looks like she's 29. And so that's a really interesting thing. And I remember I had this epiphany almost a year ago as I started to treat people, see what was going on with my mom, and see what's going on with myself. And I don't actually like to treat myself a lot. It's expensive. And actually, I think it's done things with my mind, making it too sharp and too aware. But, I did have the epiphany or this real strange, odd sense that if I didn't want to and if I could afford not to, I wouldn't have to grow old. With just 2 out of 3 of these things, the telomerase activator and the exosomes were enough to just flatten that curve out to a really long time. So uh, the senolysis is not really ready for prime time. The stem cell banking is probably ready. There are future cancer treatments using exosomes, which would be trivial to create if you wanted. You could find out what genes were missing from your particular form of cancer and put them in one of these Pfizer coronaviruses. The problem is there's no money in it. Everyone's different, bespoke personalized medicine. But in the future, if we wanted to, we could all probably treat our bodies like a Volvo P1800. We could get rid of cancers quite easily with exosome technology. We could reduce or make negligible the amount of senescence, and we could bank and replenish our stem cells at a younger age. So all this stuff is advisable, it's accessible, it's coming. We're spending trillions of dollars inciting wars and unrests in the world. If we wanted to cure aging, which arguably I would say we really don't. We want a war on aging, just like we want a war on terrorism and a war on drugs. It's really the war that generates the income for the people who are disingenuously leading it. So anyway, so that's my kind of flurry. It's things that if you've heard my lectures or seen things that I've done, you know. But I believe everything I'm saying is very, very intuitive, based on science. It's common sense. It will become trivially obvious when people someday are forced to recognize it. But I don't think aging is an insurmountable problem. It needs to be understood as a combination of many different factors. But the truism is that we all start as one cell and we're going to dust. And in the meantime, it's a pretty easy ride. The things that make people, when I ask in an audience of people why they don't want to live to 120 years, they always assume I'm going to be infirmed, I'm going to be sick, I'm going to be a burden, I'm going to lose my mind. But, if everyone looked like Adeline, 29, they would have a very, very different approach. Then they think about money. Well, I'll run out of money. But it's kind of funny. I do think that aging will be a problem that will be trivial, just like getting from New York to Paris in 6 hours. It's going to be cured by science eventually. So, with that, I'm going to end it and take questions. If anyone has questions, I will ask you to raise your hand or type into the box and we can field those. Yeah. All right. So, anybody has a question? Oh, we got a raised hand here. We're going to unmute Christine.
So Dr. Park,
Yes, ma'am.
let's say there was someone who really wanted to be aggressive about this and felt that this was a good thing to do, and maybe they were wealthy. I'm not wealthy, but let's say somebody was wealthy. How often do you think somebody should have such a treatment?
I mean, that's a great question. A colleague of mine just had a billionaire in Silicon Valley order like 70 vials. I guess you could stockpile like beans in a A-bomb bunker. Um, Nobody knows. The founder, Duncan, he put 50 vials into himself at once, and he said it helped with his deep wrinkles. But, we have to talk about the fact that we have, 7 trillion cells, but, there's probably another order of magnitude higher of exosomes, meaning like quadrillion exosomes. Of course, not all exosomes are the same. Not all words and sentences and songs and messages are the same. So maybe a little dab will do you. But to answer your question, uh, nobody knows. The people who don't pay for it, they often do 15 billion every month or every 2, 3 months. Um, but you got to be careful. I remember I treated my mom, and she blamed exosomes for getting sick. In 2019, I treated her shoulder, and she said she went to see a optician, and the tech had really bad breath and was breathing on her. Right, aloha, aloha is nearby breathing, and she got an upper respiratory infection. She lost her sense of smell, which is, of course, coronavirus 2000 whatever before it was a thing, and she blamed the exosomes. So this reminds us that a patient just wrote me today, and she wants to get treated when I come to see you in 2 weeks. And she said, "Oh, I'm getting my coronavirus vaccine." I said, "No, no, you can't take it." Like Duncan Ross said, who's an immunologist, and when he got sick, he also didn't want to blunt his immune system. So what I'm trying to say is you can't take it constantly. Your immune system is meant to go through flux, on and off, up and down. If you're suffering from autoimmunity, you probably don't want a lot of immune activation. So yeah, you could take that. But would I take it constantly, like the telomerase activator? Probably not. Um, but if money were no object and you had severe illnesses, I would treat the areas. I would treat the shoulders, the knees, whatever. Uh, no one knows the answer to your question, but probably more is more. I got chided because I went and treated people, and it's very expensive. But there's most people are charging much more money than I am, and especially if someone's been treated before, I tend to discount it quite a bit because I want them to have results and to be happy. But they said, well, you're only using 5 billion. Most people are using 15 billion. So I guess if money were no object, and we do accept cryptocurrency, then you can probably do like 25, 30 billion every month or two. But you got to be careful because we don't really know if it's linear, geometric, what the effects of shutting down your immune system will be in the long run. But we do know pulses of short regeneration are good, especially for musculoskeletal indications, but also long term. Some of the neurological, some of the brain stuff, maybe better. But it's just one tool in the armamentarium. It's just there's also the telomerase activators. There's also healthy eating and lifestyle, and there's also senolysis, which again will be part of it.
If someone was taking this aggressive approach, what ratio of IV to nasal administration would you be suggesting?
Yeah, no, that's a good question. The question is what ratio of IV to nasal? To my knowledge, I'm the only one in the world currently doing the nasal. Most people are doing the inhaled version of it, where you thaw and refrigerate for 24 hours and you sniff it in a spray bottle. It's a technique I learned when I was training for stem cells, uh, and it's pretty safe, well tolerated, and it has some biological basis. Uh, so, since I'm the only one doing it, I really don't have an answer to that. I know Deb has done a lot, and she will do the IV and the nasal. And she really thinks it's helped for different things in terms of her memory, her enjoyment of reading. So she is one of those people. I don't think she's independently wealthy. I don't know her finances, but she has committed to the idea that this is helping her, and it seems to be paying dividends. Um, so I don't know the answer to your question yet. I mean, if you give it IV, it'll go everywhere, crosses the blood-brain barrier. It's very small. Just like, if you give it in a joint, it'll leak out of the joint. That's part of the problem. And, I had a gentleman who I treated his hip. He said, "Oh, my hip is pretty much the same." Now, granted, this is very advanced osteoarthritis. He's on the schedule for replacement. He's got osteophytes, no cartilage. He said, "But my back is better." Same thing with Doug, who wants a third treatment now. He is the 82-year-old who runs 2,000 miles. And he says, "Well, you treated my knee, it leaked out, and now my thumb got better." So these things will go everywhere. But again, location is important. I think it's important to start them off where they might do the most benefit. Okay, thanks for the great questions, Christine. I'll see you in a couple of weeks.
Mary has a question.
All right, let me see if I can get over to that. Mary, there's two Marys. Mary Farrell, Mary Cummings. Oh, I'm going to scroll up here. Well, Dora asked a question. The price of the exosomes, please. The price is very expensive. It's usually $500 to $700 per billion, and even like the 5 billion, the wholesale runs almost $3,000. So, I mean the 15 billion, it's very expensive. But people think there's a huge margin in this. There really is not. Um, People will somehow, if I have a case of compassionate use, somebody with a horrible disease that is incurable, I might do it at or below cost sometimes. But yeah, I mean, there are people. One of the standard influencers has a doctor he's working with in Beverly Hills, and for 15 billion, they're getting $7,500. Uh, so everyone is all out there, but generally it's $500 to $700 retail per billion. Mary asks, is there any research being done where you could sign up to be part of a study? Uh, I do know that as of about 3 weeks ago, they got approval from the FDA to study COVID, as there was some anecdotal evidence that it was helping. I know there was a study out of Israel showing a different version of exosomes that was helping. But if you have moderate to severe COVID, I think they're doing a 48 patient trial now on mitigating that. But of course, you don't want to get COVID just so you can get free exosomes. Okay, any other questions? Uh, Lynn Davis is on. I hope everyone kind of got the gist of what I was saying with the aging. I don't think it's really that complicated. I think people try and make it complicated and they have their pet projects, and there's a lot of influencers, but I've been horribly unimpressed with the results on all these anti-aging gurus. I would say Terry Hertog, who's a big, big hormone guy, he probably looks the best, but everyone else I see at these conferences is getting older, and I don't like that. But of course, maybe the point is not to actually cure aging, but to fight the war and get funding for the war. Another question. Lynn asks, is there anything that could help with scarring alopecia? Yes, as we talked about in the alopecia webinar, Um, well, central scarring alopecia is something that is autoimmune, maybe also toxic related from hair products, very common African American women. But it basically it's the same process. You're getting a destruction of those niches of the hair follicles, and so hopefully, as we saw with the alopecia with KW, he regrew a full head of hair. Now he thinks it's all Rogaine mitigated, possibly, but I would argue that maybe there was a change in the phenotype and some healing inflammation reversal because his result was superb. And I would check that out if you were interested. If I want exosomes in both my knees and lower back, how much would that be? Again, this is like buying gas. Um, when you buy gas, it could be $2 a gallon, it could be $4 a gallon. It's $4 where they're paying more for real estate. Because I do not currently have employees, my overhead is less, and because I think it's a bit ethically challenged to charge full retail, uh, even though I'm doing stuff that other people cannot do, I tend not to overcharge. And again, if the effects are less than, unfortunately, a lot of it's self-inflicted. If a lot of times, as Duncan reprimanded me, I'm not giving enough. I'm not charging enough. But you know, if you're gonna do your knees and lower back, it's kind of like saying, how much gas do I need to buy? Well, it depends on how far you want to go and how fast. And I would think at a minimum you'd want to get 5 billion. If you want to go bigger, 15 billion is good. That'll set you back. Again, the street value of that is 7,500. We could do it for significantly less. Um, so you can contact me, Dora, offline if you'd like, and we can chat about it. Okay. Uh, any other questions from the attendees? Thank you for attending. I really appreciate it. All righty then. Okay, everybody. Well, it's a pleasure being with you, and I will edit this, and we'll see you all next time, next that channel. Okay. Okay, everybody. Thanks for joining in. This is Dr. Ed Park, completing a webinar on aging. And if you have any questions, you can email me at drpark@rechargebiomedical.com. Thanks a lot.